Theanne Griffith. I'll be talking about some work from my lab at UC Davis on mechanisms of mammalian proprioception. Okay, so a little bit about my lab. We're broadly interested in somatosensation, but what I'm going to be talking about today is the proprioception branch of our arm, the proprioception branch of our lab. And so one of the overarching questions that we're really excited to begin tackling is how sensory signaling drives developmental, adaptive, or regenerative processes within motor systems. And to address this question, we leverage various mouse models of ion channel dysfunction with a primary focus on voltage-gated sodium channels. And we're also primarily interested in proprioceptive sensory signaling, probably the most sensory, some of the most relevant sensory information that's transmitted to motor circuits. And our analyses very much span biophysics to the behavioral levels. So what is proprioception? We're going to do a little bit of audience participation. I'm going to ask you to take your finger, close your eyes, and touch your nose. That's pretty easy, right? That's because you have sensory proprioceptors that are innervating your skeletal muscle and detecting changes in muscle length and tension. And this sensory information, in essence, provides an internal spatial self-awareness that is largely subconscious and forms this connection between muscle and mind. And so, as I mentioned, proprioception is mediated by these proprioceptors whose cell bodies are located in the dorsal root ganglion and send axons to skeletal muscle and tendons that detect changes in muscle movement. They transmit this sensory information to motor networks in the spinal cord. Proprioception is incredibly important for all kinds of movement, whether it be kind of complex movement that most people in this room might not be able to do, or simple things that we can all do, like walking, maintaining posture, and also proprioceptive training has been used for a long time by physical therapists to accelerate functional recovery after injury. So at the cellular ion channel level, upon muscle movement, you have opening of piezo2 ion channels, which leads to influx of calcium and sodium, provoking a receptor potential that then activates voltage-gated sodium channels, leading to signal propagation. So in my lab, we've largely focused on the differential roles that voltage-gated sodium channels have in contributing to proprioceptor activity. So using ex vivo muscle nerve recordings, we isolate out muscle and record from proprioceptive input into that muscle. And upon muscle stretch in normal control conditions, you have this robust and kind of consistent proprioceptor activity during sustained muscle stretch. However, in conditional knockout models in which sodium channels are selectively deleted in somatosensory neurons, you can get moderately impaired proprioceptor activity by targeting NAV1.1. And conversely, you can get a complete loss of proprioceptor activity by targeting NAV1.6. And I'd like to point out that the role of NAV1.1 is particularly important for maintaining excitability during static muscle stretch, whereas the role of NAV1.6 is just generating electrical signals, whether it be static or dynamic muscle movement. These electrical changes at the cellular level are accompanied by clear behavioral phenotypes at the organismal level. And so you can see here when you hold either an NAV1.1 or an NAV1.6 conditional knockout mouse by its tail, you have very, very clear and robust behavioral deficits, motor deficits that we've quantified and published. And most recently, and also I should say, if you kind of have, if you're used to looking at mouse models of behavior, there are some differences in the behavioral phenotypes, similarly to the differences that we see at the cellular level. And we think that these differences might come down to something as simple as channel localization. So we were able to localize both NAV1.6 and NAV1.1 in those muscle spindle proprioceptor end organs. And what we found was really interesting. NAV1.6 is pretty much restricted to these tiny little clusters, 100% of which we found were positive for ANG-G, a marker of axon initial segments, whereas NAV1.1 had this more diffuse pattern that is consistent with its role in amplifying signals for consistent and reliable firing. And this is just a kind of up-close version of that, where you can really see the two channels occupy different cellular regions within proprioceptive end organs. And again, we think this contributes to their differential role in encoding mammalian proprioception. But what I want to talk to you about today, that was all kind of a summary of some published work. What I'd like to talk to you today about is the spinal monosynaptic reflex. So this is perhaps one of the most simple circuits within the mammalian nervous system, but incredibly, incredibly important. And we've probably all had this reflex tested when we go to the doctor. The doctor will tap your knee with the little mallet here. That will activate those proprioceptive endings in your hamstring. They will then activate monosynaptically motor neurons that reside in the ventral horn of the spinal cord that are also innervating that muscle, leading to its contraction. And at the same time, disynaptic and polysynaptic circuits are going to inhibit motor neurons that are innervating the synergistic muscle. I said this is a quadricep, this is the hamstring, sorry. And leading to its relaxation, right? And so the end result is this upward movement of your leg. And although it's very simple, it's really essential for motor reflexes and also prevents excessive muscle stretch during purposeful movements. And so a project scientist in the lab, Chetan Nagaraja, was really interested and intrigued by the organization of this monosynaptic reflex circuitry. So as you may know, our spinal cord is divided into regions: cervical, thoracic, and lumbar. And within those regions, you have different spinal segments that align with your vertebrae. So here, I am showing spinal segments lumbar 4 and lumbar 5, abbreviated L4 and L5. So kind of traditional thought is that this monosynaptic reflex arc is designed segmentally, whereby proprioceptive input from L5 only innervates motor neurons found in L5, and similarly, proprioceptive input from L4 would only innervate motor neurons found in L4. But Chetan was really curious as to whether or not there could be any kind of intersegmental communication, especially that might occur during development, and to, generally speaking, what extent does this reflex arc undergo refinement during postnatal development? To address this question, he used a hemisected spinal cord preparation with 3 suction electrodes. One electrode was hooked up to the dorsal root from the lumbar 5 segment containing those proprioceptive sensory axons. And then he had a suctional recording electrode hooked up to both the ventral root at L5 and the ventral root at L4 to record segmental and intersegmental responses, respectively. So when he did these recordings across different developmental time points, he indeed found, in addition to that nice, robust segmental response, an intersegmental response at early developmental time points during that first week of postnatal development that was rapidly diminished by the end of the second week of postnatal development. And here's a quantification of those data right here. We have the raw amplitude data as well as showing that magnitude of the intersegmental response being high in early development. Now, we think that this is in fact a bona fide monosynaptic connections for 2 primary reasons. One, when we look at the latency of the response within a developmental group between the L4 and L5 responses, we see that they're not significantly different and in general had a range of 0 to 300 microseconds, with disynaptic synaptic signaling requiring at least about 2 to 2.5 milliseconds. So it's much, much faster than what you would see in a disynaptic circuit. And additionally, we also conducted some tracing experiments whereby Chetan filled the ventral root L4 to label L4 motor neurons with a dextrin conjugated to 1-fluorophore, and then filled that dorsal root 5 segment with a different fluorophore, and found that at P5 you saw really robust innervation of motor neurons in the neighboring segment that was really largely diminished by even P9. And I wanted to note that although we see that almost every single motor neuron received at least 1 synaptic input, or I should say motor neuron in L4 received 1 synaptic input from L5, we only are seeing, about a 30% or so magnitude of that intersegmental response, suggesting that a lot of these are weak or perhaps non-functional synapses. And so we wanted to see if this kind of refinement was activity dependent, because that's a phenomenon in the developing nervous system. Weak synapses are removed in an activity dependent manner. And so we went back to our model of severely impaired proprioceptor activity, this NAV1.6 conditional knockout model. And just a reminder, in this model, NAV1.6 is only absent in somatosensory neurons. It's still present in motor neurons in the spinal cord and pretty much everywhere else in the nervous system. And so what Chetan did was a nice thorough analysis, breaking it down by age group. and recording just segmental responses to confirm that indeed these synapses were impaired. We had previously shown that after the onset of walking behaviors at P14, these responses were gone. So here he's doing a much more careful analysis and found that indeed from P8 to 10 or P11 to 13, that segmental response had a longer latency and required a larger threshold despite having similar amplitudes. And again, he found that by P14, the response was gone. So this shows that during development in this model, this synaptic reflex, this monosynaptic reflex arc does have abnormal synaptic activity. So he then asked, does this abnormal synaptic activity lead to a persistence of this intersegmental response? And that's exactly what he found. So very similar to the experiments I showed you previously, in flux conditions at P8 to 10, you have a pretty small intersegmental response that is completely gone by P11 to 13, yet this clearly persists in our conditional knockout models, despite, and again, we think these are monosynaptic based on our latency analysis. So this shows that this refinement of these intersegmental synapses is indeed activity dependent and at least depends upon NAV1.6 in proprioceptors. We wanted to get at the cellular and molecular mechanisms that might underlie this refinement. And so in the developing nervous system, microglia are often responsible for identifying and engulfing weak synapses. And so we thought to quantify microglial counts across development. And while we did indeed see within the ventral horn of the spinal cord, again, that region where you have all of your motor networks, we did see this increase in microglial counts from P5 to P9 that stabilized by P12. When we looked at P9 spinal cords from either floxed or NAV1.6 conditional knockout mice, we saw no difference. So the difference in microglial numbers is not contributing to this synaptic refinement, or lack thereof in our NAV1.6 conditional knockout mice. So we thought to look a little bit upstream and focused our attention on the complement signaling pathway. So during development, the complement signaling pathway, which is part of both of our innate and adaptive immune systems, tags synapses, usually weak synapses, for future engulfment by microglia, as you see here. And we're particularly interested in one protein in this pathway, C1qa, as this protein has been shown to be increased in ALS, which is a motor neuron disease. And what was priorly shown was that you have this increase in C1qa expression that leads to over-pruning of proprioceptive synapses onto motor neurons, contributing to disease pathology. So we were curious, in our case, if the reverse was true, if you had perhaps lower levels of C1qa in our conditional knockout model that could explain this kind of persistence of these exuberant synapses. And that's what we found. So when we labeled the ventral horn again of either floxed or NAV1.6 conditional knockout mice, we saw significantly less C1qa signal in our conditional knockout mice compared to our flox controls, as shown in magenta here. So this suggests that somehow C1qa is not being expressed as highly in the developing spinal cord in the absence of normal proprioceptor activity. So we wanted to kind of tie some functionality into this, and so what Chetan did is he got his hands on a full knockout of C1qa and repeated some of these intersegmental electrophysiology experiments. And so he first wanted to ensure that in the model, everything was working as we had seen in the 2 prior lines. And indeed, we were able to, at P4 to 7, record this robust intersegmental response. But interestingly, just the segmental response in and of itself was significantly larger in these C1qa knockout mice, consistent with the notion that in these mice, you don't have effective synaptic pruning. However, the amplitude at this age was not, the magnitude of the intersegmental response at this age was not different between control and knockout mice. Again, though, we do see this increased threshold in the knockout mice compared to the control, suggestive of additional synapses. And if we just look at the latency of the segmental response, we do see this significant decrease. Again, supporting the idea that in early development in the C1qa knockout mice, you have exuberant synapses. When we look then at P11 to 13, Chetan observed exactly the same thing that he observed in his NAV1.6 conditional knockout mice. Whereas control mice at this age had no intersegmental response, there was still a robust intersegmental response in the knockout mice, implicating a role for both NAV1.6 signaling and C1qa signaling in the refinement of the monosynaptic reflex arc. So to kind of summarize what I've shared with you today, prior to postnatal day 10, you do, in fact, have intersegmental innervation of proprioceptive axons onto motor neurons in neighboring segments. Under normal conditions, you have tagging of those proprioceptive synapses, likely by proteins like C1qa in the complement signaling pathway that leads to their removal, such that by the end of the first postnatal week, you have no more intersegmental synapses and are unable to invoke an intersegmental response. However, in the absence of NAV1.6, just in proprioceptors or just in sensory neurons, that doesn't happen. You have reduced C1qa tagging of proprioceptive synapses and increased transmission, resulting in the persistence, anatomically at least, of these intersegmental synapses. And one really interesting question is, what is that activity-dependent signal that's coming from proprioceptors that induces this C1qa expression in the spinal cord? So I know we're at an ion channel meeting, and I didn't really talk about like ion channel biophysics, but I still think this has an important relevance to NAV1.6 channelopathies. So we know that NAV1.6 is essential for proprioceptor function, and we know that NAV1.6 channelopathies are often accompanied by motor deficits. However, how proprioceptive signaling is affected by sodium channelopathies, 1.6 for example, is not known. No one has done that experiment yet. And it's possible that these anatomical abnormalities in spinal cord circuits could contribute to the motor dysfunction in patients with these different NAV1.6 channelopathies. And we think that future work will very much help advance our understanding of sensory contributions to motor dysfunction in these neurodevelopmental disorders. And so with that, I would just like to thank the people that were involved. Some of the early work that I showed in the very beginning about different roles for sodium channels in proprioceptor function was spearheaded by Cyrus Espino, who is a recent graduate from the lab, a PhD student who's now going to be starting his postdoc soon. And the majority of the work that I showed you today was led by Chetan Nagaraja, a project scientist in the lab, with some help from Serena Ortiz and Akash Murali, who are as a grad student and undergraduate student in the lab, respectively. I'd like to thank you all for your attention, as well as funding sources.
It's a really interesting talk. Since C1q is the most basic way of self versus non-self discrimination, what's the idea for what's the marker that it's smelling on the synapses to be eliminated?
Yeah, I think that's a great question. We honestly don't know. The reason that we looked at C1qa is, again, because it had been shown to be upregulated when you get excessive pruning of synaptic connections between proprioceptors and motor neurons. But we honestly don't know exactly what the signal is that is driving its increase in expression. I think that's a really provocative question because, again, we're only manipulating sodium channel function in proprioceptors, and also at this time point, it's not that there's no transmission. That happens, that complete shutoff of proprioceptive transmission happens after P14, during the onset of walking behavior, so it's really just kind of impaired signaling, and that alone in and of itself seems to be insufficient to get that upregulation. So it's a great question. We don't have an answer at this time.
There's no autoimmune C1q mediated dementias or anything?
Ooh, I haven't looked into that. Yeah, that would be very cool. I haven't looked into that, so thank you for that. I will.
Great talk. I had a question. So obviously, using the C1q knockout mouse, you were able to demonstrate that you retain these cross-sectional synapses. Did you ever look at a motor or proprioceptor?
We didn't, and we still have the mouse in the lab, so we definitely could. I will say, there is nothing as obvious as what we see in our NAV 1.1 or NAV 1.6 conditional knockout mice, but that doesn't mean that they don't have more subtle deficits.
That's really fascinating talk. I was wondering whether just general increased activity, not necessarily mediated by NA 1.6, is sufficient for this activity-induced pruning.
I'm actually really curious about that myself, and that kind of gets to this question of the channelopathies, NAV 1.6 channelopathies being accompanied by motor deficits. So most NAV 1.6 mutations are actually gain-of-function mutations, and we have a loss-of-function model. And so whether or not, we don't really know, again, what proprioceptor activity looks like when you have a gain-of-function sodium channel. They already fire really fast, and so it's very possible that you could get some kind of depolarization block with a gain-of-function mutation that could end up just mimicking a loss-of-function mutation, because the motor deficits that you see in these patients are very similar to what you see in our knockout animals, and also just kind of some of those systemic effects. So it's unclear, but I think it's a really, really interesting experiment. And if we could get our hands on either a point mutation mouse or a conditional, I would really love to have a conditional mutant knock-in, where we could just knock in that mutation in sensory neurons or in motor neurons and kind of really figure out the spatial landscape of what a human disease-causing mutation is doing in a cell type specific manner. All right. Thank you.